During a woman's reproductive years, the ovaries and uterus go through a series of histological changes approximately every 28 days. This rhythmical pattern is referred to as the female reproductive cycle or the menstrual cycle. The histological changes which occur to these organs are a result of a rhythmical pattern of secretion of the two pituitary gonadotropins, FSH and LH, and the ovarian hormones, progesterone and estrogen. Simply stated, it is the pituitary gonadotropins that act upon the ovary and the ovarian hormones which regulate the cyclic changes that occur in the uterus. This first graph shows the relative changes in the concentration of these four groups of hormones, along with a diagrammatic representation of the histological changes which are occurring during the cycle. The onset of menses is the first day of the ovarian cycle. For convenience, the cycle has been divided into two phases. The first one half is referred to as the follicular phase and is characterized by the growth of the Graafian follicle in the ovary. The second phase is the luteal phase, and it is the time during which the ovary develops a corpus luteum. Ovulation usually occurs midway in the cycle or approximately on day 14. During the early part of the follicular phase, there is a slight increase in FSH, which is responsible for the development of a Graafian follicle. The Graafian follicle, in turn, is the site of estrogen secretion. Therefore, as the follicle grows, the concentration of estrogen in plasma increases. The main action of the estrogens is to stimulate the rebuilding of the endometrium, which was lost with menstruation. Estrogens have a negative feedback on FSH, and the concentration of plasma FSH decreases. The secretion of FSH and LH rapidly increase. Approximately 1 day before ovulation, the reason this occurs is not understood, but it is known that LRF, luteinizing releasing factor, is released from the hypothalamus and produces a surge in these gonadotropins, especially in LH, which brings about ovulation. Ovulation involves the release of the fully developed egg from the follicle into the fallopian tubes of the uterus. Following ovulation, the follicular cells then change into a structure referred to as a corpus luteum under the influence of luteinizing hormone. The corpus luteum is a site of production of both estrogens and progesterone, and thus you see a rise in these ovarian hormones. The ovarian hormones have a negative feedback on FSH and LH as evidenced by the decrease in secretion of the gonadotropins. The estrogens and progesterone complete the rebuilding of the uterine endometrium and prepares it for implantation. Progesterone also has a stabilizing effect on the endometrium and helps maintain it by inhibiting uterine motility. If fertilization does not occur to the ovulated egg, the corpus luteum will regress, and the secretion of progesterone and estrogens decrease. With the drop in progesterone, the endometrium cannot be maintained, and menses occurs, and the next cycle begins. If fertilization does occur, then the pattern of secretion of these four hormones changes. Graph 2 shows the changes in concentration of the ovarian hormones and the gonadotropins during one complete cycle, followed by another cycle which is interrupted by fertilization. Fertilization usually occurs within 24 hours after ovulation, and it usually occurs in the upper 1/3 of the fallopian tube. The fertilized egg then requires approximately 3 days for its passage through the fallopian tube into the uterus, where it floats freely for another 4 days. Implantation results from the action of the trophoblast cells on the blastocyst. Later, these trophoblast cells, along with endometrial cells of the uterus, will form the placenta. At the time of implantation, day 7 or 8 after ovulation, the trophoblast cells are secreting enough of the hormone, HCG, that it can be detected in the mother's body fluids. This hormone is the basis for early pregnancy test. HCG's pattern of secretion is shown on this diagram. As you can see, its peak secretion is approximately 6 weeks following ovulation, or the 6th week of pregnancy. This is followed by a rapid decrease in secretion. This protein hormone is secreted in large quantities during the first trimester. The biological action of HCG is very much like that of LH. Its function is to prevent the involution of the corpus luteum, and thus ensures high circulating levels of both estrogens and progesterone. These two steroids in turn function to 1. Maintain the endometrium, and 2. Inhibit the release of FSH and LH, and thus prevent the development of the primary follicles in the ovary. By the end of the first trimester, the placenta is well established and is secreting large quantities of estrogens and progesterone. Thus the corpus luteum is no longer needed, and it involutes due to the decrease in secretion of HCG. In 1927, 2 German gynecologists published a method which could be used to diagnose pregnancy within 6 weeks after fertilization. They collected urine from a female. This was concentrated and injected into female immature rodents. 96 hours later, the rodents were sacrificed, and the ovaries and uterus of the rodents was examined. The uterus and ovaries were examined. If human urine contains no HCG, the test is negative. In other words, there is no corpus luteum, no endometrial or myometrial uterine growth. If HCG is present in human urine, there will be a corpus luteum in the rodent's ovaries, and there will be uterine growth of the rat's uterus. Therefore, the test is positive, and the human female is pregnant. In 1960, Dr. Persons and Yao, 2 American endocrinologists, developed a sensitive and rapid method for detection of many hormones in blood and urine. In 1976, they shared in the Nobel Prize for this work. This test consisted of a immunological test for pregnancy, and this was developed between 1968 to 79. Basically, the protein, the glycoprotein, human HCG, is collected and purified. It is injected into rabbits. They form an antibody, anti-HCG, against it. Then, if the HCG is present in the female's urine, an antibody collected from rabbits is added to it, agglutination will occur. The immunoassay for HCG is easier to perform, and it is very sensitive. Therefore, it can be used to detect pregnancy soon after fertilization. However, this method is not as accurate as the bioassay for HCG. This is a test which can be purchased in a drugstore. A sample of urine is collected in the morning, and it should be used fresh and not stored. The cap is removed to expose the lyophilized antibody. Distilled water is added to dissolve the protein antibody. Urine is removed, and 3 drops are added to the solution. The mixture is shaken. The initial appearance of the mixture is a brown homogeneous solution. If the urine contains HCG, A precipitate appears in the bottom within 2 hours and is a positive test for pregnancy. In summary, if this test is purchased and used in the home, you should be aware that a negative test is inconclusive and should be repeated, especially if the onset of the menses is delayed. Regardless of the outcome of this test, a physician should be consulted if the female suspects pregnancy or a disorder of the ovarian cycle. Perhaps a more realistic title for this TV tape could be called, "Will She Tell You If She Knows?"